we'll start. Welcome to today's World Potato Congress webinar. I'd like to start by reminding you of the next World Potato Congress due to take place in Adelaide on June the 23rd to 26th, 2024, and encourage you to register for that meeting. Thanks for attending. Please submit any questions that you have about the talk into the QA tab, and I will come to these at the end of my presentation. And a recorded version of the webinar and also a PDF will be made available on the website afterwards. I'd also like to thank the sustaining partners of the World Potato Congress for sponsorship, and that allows additional activities such as these webinars to take place. And finally, before I begin, I'd like to draw your attention to the upcoming EAPR Pathology and Pest section meeting taking place in France at the beginning of September this year, and encourage you to look at the website, and registration is now open for this meeting. So the title of the presentation is Potato Tuber Blemish Diseases, Status and Management. And by way of introduction, I'm Alison. I'm a potato pathologist located here at the James Hutton Institute in Dundee in Scotland. And just for information, shown on the map in green are the main potato growing areas in Great Britain. So in today's talk, I'd like to cover a few aspects of potato blemish diseases, focusing on what they are, whether they matter in terms of losses due to reduced yield, and then think about what we already know in terms of some of the epidemiology, diagnostics and control options. I'll try to give examples from some older research that we carried out here, and some links to useful resources and more recent findings from other researchers. So I think it's safe to say that these diseases are not particularly extensively studied. They're not one entity, so it's quite difficult to group them and speak about them collectively. They tend to be most problematic for the washed pre-pack market, particularly if being stored. And impacts on quality and yield are very variable between the diseases, and similarly, control measures vary between diseases and between countries. So I think the next slide illustrates some of the diversity that I'm referring to here. So here are just some of the more common diseases that we think of under the potato skin blemish umbrella, ranging from those which are almost exclusively found on tubers, such as common scab, silver scurf, and skin spot, and those which are also able to cause other symptoms. For example, rhizoctonia causing stem and stolon canker, as well as black scurf, shown here. Spongospora causing both powdery scab and root galling, and black dots causing disease on both stems and tubers. And also a case here of several of these pathogens coexisting on the same tuber. So I will focus mainly on black dot in this talk, with some information about silver scurf where it's available. For the identification of potato tuber blemish diseases and more detail on a range of pathogens, I refer you to this useful online resource, as today I'll only really be able to scratch the surface of some of the diseases. So I wanted to mention that tolerances for skin blemish diseases vary between countries and for different purposes. So please make reference to your own national rules for inspection and export when thinking about allowable levels of disease or those that have a potentially economic impact. Blemish disease tolerances are often grouped together for this purpose, as illustrated here, with individual tolerances, group tolerances, and collective group tolerances. So please refer to your local rules. As I said, I'm going to focus mainly on black dot, but note that there are 3 previous World Potato Congress webinars available where you can find information specifically about powdery scab, about rhizoctonia diseases, including black scurf, and seed management practices. And I recommend that you look at these if you're interested. So if we start with some general information about silver scurf, it's an important blemish disease with a worldwide distribution. The pathogen, Helminthosporium solani, is seed-borne, and the disease is not generally associated with yield loss, although it can cause shriveling and water loss from the tubers. Infection occurs in the field and can develop during storage, particularly under humid conditions. Seed treatment is an effective management strategy in contrast with black dot. And again, I refer you to the earlier presentation of Gary Secor in this respect. There are relatively few alternative hosts, mainly solanaceous species, and high levels of resistance have not been identified, although varietal differences have been seen, and I'll come back to this point. So black dot is also an important blemish disease with a worldwide distribution. In the past, and possibly even now, black dot was underreported, as it was often confused with or grouped together with silver scurf. Disease development and severity reports vary. For example, in the UK, we don't associate Colletotricum cacoides infections with yield loss, whereas researchers in the US and Israel have reported losses of up to about 30%, but the results can be inconsistent. C. cacoides is also implicated in the potato early dying complex alongside verticillium and nematode species. So C. cacoides has numerous alternative hosts, some of which are listed here, and include solanaceous and non-solanaceous species. Work by Nitsan et al. Also lists a range of rotational crops as hosts for cacoides. And I noticed recently that there's a new disease report for black dot on sugar beet in the US. So how prevalent is black dot on seed and in the soil? So earlier work of ours from the UK found that 81% of 122 commercial soils in potato production were contaminated when tested using diagnostic assays. And similarly, 76% of seed stocks of various source and variety, and 25% of seed stocks were contaminated but visually symptomless. Data from a Norwegian survey found that there was more than a 50% incidence of tubers with black dot, sampled from various regions of the country. And this work also agrees with earlier work in France and the US that found using baiting assays that 80 to 100% of field samples were contaminated. So it's fairly widespread. Identification of black dot has sometimes been an issue. So it's caused by the pathogen Colletotricum cacoides, takes its name from the tiny jet black microsclerotia. Symptoms can be present on stems, stolons, roots, and tubers, and there's a significant literature on black dot development on plants. Tuber symptoms are present at harvest. The affected skin is initially light brown to unblemished with dots, dark brown patches can develop, and lesions tend to be irregularly shaped, as you see here. Some lesions, particularly those on red skin potatoes and well-developed lesions, look silvery, leading to confusion with silver scurf. Hyphae of cacoides are found in the periderm, but it doesn't appear to penetrate the cortical cells, and there's work from Masana Kadina that goes into more detail on that aspect. In contrast, silver scurf is caused by Helminthosporium solani. Small silvery grey spots can grow and merge under moist and warm conditions, and during storage the colour may change to a darker grey or brown. To confirm the presence of silver scurf, look for short black threads at a magnification of ×10. And here you see the distinctive conidiophores seen here in these great pictures on the surface of a tuber. And here at the bottom, you can see the typical spores in another great image from Viviana. Silver scurf may be confused with black dot. They frequently occur on the same tuber, but they rarely overlap. So silver scurf and black dot are shown here at the same magnification, just highlighting the differences that allow identification. So there's quite a lot of literature describing colonization of plants by Colletotricum, but in brief, spores spread by the wind, rain, and irrigation germinate in the presence of water and directly penetrate the plant tissue. C. cacoides infects all below ground organs as well as stems and leaves. And in potatoes, roots, stolons, and daughter tubers are quickly colonized, showing symptoms of black dot soon after their emergence, while stems show disease approximately 7 to 10 weeks after inoculation. There are multiple disease cycles during crop growth as spores spread and infect neighboring plants. And the characteristic black dot symptoms or sclerotia develop on the tuber, and these provide a means for the pathogen to overwinter on tubers and plant debris, produce spores, and start the cycle again. So I just wanted to mention here that in the next section of work that I'm going to talk about, quantitative real-time diagnostics have been used as a tool for investigating the epidemiology of the diseases, both in our work and that of others. These are now really quite old assays, but still effective, and many of the assays are published. In Australia, the Predictor PT soil test is available commercially, and this can give a good indication of disease risk on progeny tubers based on inoculum levels in the soil. And I'll come back to the question of setting such thresholds. There are also various methods published for extraction of DNA from tuber peel sap and from soil. So what are diagnostics good for? Well, they're useful when symptomless infections may be present on seed or on progeny going into storage. And they also enable us to quantify soil-borne inoculum to evaluate disease risk. It seems really obvious to say that the assays will only quantify what is in the sample taken. But this is an important point, as the sample must represent the seed stock or the field as a whole. And sampling strategies therefore underpin the ability of diagnostic assays to predict risk. In the two grids here, you can see the distribution of soil-borne inoculum of Colletotricum cacodes in two different fields, field A and field B, that we tested using the diagnostic assay, and how it differs and the chance of missing inoculum, for example here, if too few subsamples are taken, as it's patchily distributed. Similarly, for tuber sampling, whether for visual disease assessment or diagnostic testing, results can be biased by cutting corners with sampling. And so there are some guidelines which should be adhered to. Samples should be taken from as many boxes as possible. Sampling from the top of boxes will result in bias. Avoid handling rotting tubers and avoid cross-contamination. So here's some old work of ours looking at the role of seed-borne inoculum in causing black dot on progeny tubers. We planted seed with different levels of disease ranging from visually clean to more than 20% surface affected with black dot symptoms into Colletotricum-free soil under controlled environmental conditions. And we found no differences, seen here, in disease incidence on the progeny tubers relating to the levels on the seed that was planted. And this indicates that levels of seed-borne inoculum are not predictive of black dot disease risk on the progeny tubers. So we then showed the same thing in field trials planted with two potato varieties. Again, the incidence of black dot did not increase with increasing seed contamination. It did, however, vary according to the variety, with more disease being seen consistently on Maris piper, a more susceptible variety shown here in green, than on variety Sante, which is more resistant. So by looking simultaneously at the effect of seed and soil-borne inoculum on disease, here we can see that the seed data that I just showed you and the lack of correlation with disease. If we now look at the data from different levels of soil inoculum planted with seed with no visual contamination here, we can see that as the level of soil inoculum increases, so does the incidence of black dot on the progeny tubers, and that disease levels are much higher. So in this graph, contamination as measured by amount of DNA is used as a proxy for disease, but actually there's a very strong relationship between those two sets of data. So it begs the question of whether seed-borne inoculum or black dot on tubers is of any significance whatsoever. If you accept that blemishing is a real economic and marketing issue in the first place, then it does matter, because the contaminated seed can cause disease on the progeny tubers, enhancing the major effect of the soil inoculum, and can infest the soil once planted and perpetuate and spread the pathogen. So we know that real-time PCR detects and quantifies the amount of target pathogen within a sample, and it's a sensitive, specific, and reliable technique. We can quantify levels of soil inoculum, and I've just shown that there's some sort of relationship between inoculum and disease. But ideally, this could be used as a tool to allow growers to make decisions on site selection, varieties, and control options based on the severity of disease risk. And so this was the next step in the equation. By monitoring soil inoculum of Colletotricum in 125 commercial soils planted with a range of varieties at a range of locations and under commercial management practices, and assessing disease on the progeny tubers, we were able to assign disease risk categories, seen here as low, medium, and high risk, according to soil inoculum levels pre-planting, measured as DNA per gram of soil. So other researchers also did this exercise later in their own countries and also found good relationships, although the levels of pathogen detected and the varietal susceptibility can vary between countries, and the risk categories may have to be modified accordingly. So in similar and more recent work on silver scurf, Masana Cadena et al. Found that silver scurf disease incidence here and severity here did not correlate with disease severity on the progeny tubers at harvest, although in this case it was the main source of inoculum. Soil inoculum was not detected in their trials or in any of our earlier work, and it's known that survival of Helminthosporium solani in soil is relatively short-lived, especially compared with black dot, and can be controlled therefore by good rotations. One main way of controlling plant diseases, and the cornerstone of IPM approaches, is the use of resistant varieties. The planting of more resistant varieties will reduce the risk of black dot. However, resistance ratings are not often available for national varieties. Qualitative resistance has not been reported, and commercial varieties show different levels of susceptibility or quantitative resistance. Differences in resistance are often, I think, coincidental rather than having been deliberately selected for as part of a breeding program, as other targets such as late blight or potato cyst nematode have precedence. However, differences in varietal resistance do exist. Here we have work from Masana Cadena in Switzerland, showing a range of resistance ratings for their varieties to black dot. And similarly, these results from 6 of our trials conducted at 2 locations over 3 years show that the percentage of unmarketable tubers, excuse me, defined here as the percentage of tubers with 10% coverage or more of black dot, is significantly reduced when more resistant varieties are grown. For example, from 6% of unmarketable tubers at a resistance rating of 4 on a 1 to 9 scale of increasing resistance, compared with less than 1% at a resistance rating of 7. In other work looking in more detail at mechanisms of resistance, Masana Cadena recently showed that susceptibility was not correlated with skin thickness, and that metabolites with antifungal properties may have a role. Excuse me. Here we can see that the effect of variety on disease development appears to be greatest on tuber black dot compared with stems, roots, and stolons at a series of sampling dates from 105 days after planting, and again reflects the higher resistance levels seen in variety Saxon, here in purple, compared with Maris Piper in green. The Swiss researchers also measured varietal resistance to silver scurf in the same range of varieties, and again found differences. The ratings were found to be stable across years for most of the varieties, and they found that there was no relationship between susceptibility to silver scurf and to black dot. So these graphs, again from Masana show that all below ground tissues were colonized by Colletotricum cacoides. Infections in the roots and stolons appeared very early in the season, here and here, and their incidence remained stable throughout the growing season. Daughter tubers were infected with Colletotricum soon after tuberization, with at least 40% of the tubers infected at 70 days after planting, and between 60 to 80% at 100 days after planting and thereafter. DNA of Helminthosporium solani was found in fewer than 20% of root samples throughout the season, and fewer than 10% of stolons showed infection until 76 days after planting, after which a linear increase of Helminthosporium solani infected stolons was observed. Similarly, fewer than 15% of tubers were infected with H. solani before 92 days after planting, increasing to 40% at 100 days after planting, and reaching 40 to 80% of tubers infected at harvest. During all 3 years, the number of tubers infected remained below 50% until haulm destruction, and increased to approximately 71% at harvest. So there's also a clear effect of crop duration on black dot, with greater risk of black dot the longer the crop is in the ground. Of particularly high risk is a harvest date after early October under our growing conditions, or where crops are grown for a long duration. But the absolute date of harvest is less important than the crop duration. So here you can see that there's a consistent effect across 6 field trials of an increased percentage of unmarketable tubers at the later harvest, shown in purple, compared with the earlier harvest, shown in blue. There's a strong relationship between black dot severity here and crop duration, particularly at high levels of soil inoculum. There's still a relationship with crop duration and disease at lower levels of inoculum, but it's less pronounced. We also observed an effect of temperature and moisture on both disease incidence here on the left and severity on the right, with warmer and wetter soils here, increasing the risk of black dot under controlled environmental conditions. These factors are somewhat difficult to manage in the field, and of course, irrigation use must be balanced both with the needs of the crop itself and the potential effects on other diseases such as common scab, which can be controlled with precise use of water, and powdery scab, which can be exacerbated. When looking at the effect of azoxystrobin fungicide applied in furrow at planting on the incidence of black dot on tubers at various sampling times, we showed a significant effect of the fungicide in controlling disease from 125 days after planting and up to the final harvest. You see here the Amistar minus treatments with more disease than the Amistar plus treatments. There has been work on post-harvest storage, although I'm somewhat less familiar with this aspect. Tubers can be infected in the field or post-harvest, and store hygiene is important to reduce the risk of contamination, particularly for silver scurf. Symptoms can develop in storage, and risk can be modified by manipulating storage conditions. I'm aware of some ongoing work at Cranfield University in the UK to develop a model to predict black dot in the supply chain and to look at ethylene supplementation on black dot, but I don't have any further details or results of that work to share with you. So there are several barriers to the effective control of blemish diseases in general, relating to long-term survival in the soil, which makes rotation useful but not totally effective. That's particularly in the case of black dot, also powdery scab actually. Black dot has been shown to survive for at least 8 years or more in the soil. A limited number of effective fungicides. I've deliberately not named any fungicides here, as that will vary nationally and should be consulted upon locally, really. Some pathogens have a long latent period and a wide host range, and disease severity is unpredictable and dependent to a large extent on the weather. So for skin blemish diseases, it's important to understand crop risk in order to ascertain whether control is financially worthwhile. There are some general guidelines for factors important in controlling a range of diseases listed here, which you may wish to refer back to later. Generally, most of these factors are common sense for minimizing disease, such as the use of healthy seed, long rotations, use of varietal resistance, and trying to minimize tuber damage. And as always, we should be looking towards an IPM approach to controlling any disease. And if we look at this IPM wheel here, well, I've talked about monitoring and thresholds. Chemical control is a possibility, as is cultural control through the use of host resistance or agronomic measures such as rotation or weed control. Biological control is less well established for the blemish diseases and actually many diseases of potato in general, although there is some research in that area in terms of in-store control of blemish diseases. For black dot specifically, we know that soil inoculum levels are related to disease risk, that seed inoculum is relatively unimportant, although it does play a role in disease. Cultivar resistance significantly reduces disease. Irrigation can increase black dot disease and severity, and in-season or in-furrow fungicides can reduce tuber disease. Delayed harvest significantly increases disease, particularly at high inoculum levels, and long rotations can reduce soil inoculum and are in any case a good idea for lots of reasons in potato cultivation, and potential hosts could be managed. So these IPM approaches fit in with the mission of IPM at Hutton, which is a working group at the James Hutton Institute, working towards developing IPM strategies across a number of crops, including potatoes. I've added some of the key references to the presentation so that you can refer back to them if you wish to. And with that, I would like to draw to a close, remind you once again that the recording will be made available and also a PDF, and invite you to submit your questions in the Q&A tab. And I'll now open up the Q&A tab and see what's in there, if you give me a moment. So there's a comment that some areas of India are badly impacted of dry rot, and they're trying biocontrol agents. How can we manage emerging potato pests and disease in resource limited scenarios? Well, I think some of the control measures that I've talked about there, clean seed, of course, is a very important one. Managing risk, looking at varieties really, I think, where resources are limited, if fungicides are not available, and if possible, looking at storage conditions and date of harvest, actually. So I'm just scrolling down. Do you have verticillium wilt in the UK? In North America, black dot is increasingly treated as part of the potato early dying complex. Actually, in the UK, not so much. We expect that may well increase with warming temperatures. So verticillium wilt does occur in England. We've seen less of the potato early dying complex. I know that it is a problem, particularly in warmer countries, and actually in some Scandinavian countries, it's becoming more of a problem. So we haven't looked at that ourselves in any great detail, but I know that it is a problem and could be an increasing problem. Of all the potato diseases in the UK, where do blemish diseases rank in terms of importance? Well, it depends what your market is, I would say. So in terms of importance for crop loss, I would say that they are fairly low importance, with things like late blight being very high on that list because it's devastating. You can quickly lose your crop within a week, whereas that doesn't happen with potato blemish diseases. In terms of marketability, it can have a large impact for pre-packed potatoes. So I would say in general, quite low, but for certain markets, it is a big problem. How can I get the PDF? Well, the PDF will be available on the website alongside the webinar. Any idea about the use of plant botanicals or biopesticides against tuber diseases? Yeah, I'm not sure whether I missed that. So I briefly mentioned that. I know there's some quite old research using various essential oils for control of blemish diseases in stores. I think actually formulation was a problem. They were effective in some cases. I haven't done any work on biopesticides against tuber diseases. I think often the targets are those more important diseases or more devastating diseases like late blight, but actually it's very difficult to have a large impact on a disease like late blight, whereas a reduction in a blemish disease might be possible using these biopesticides, and particularly where fungicides are not used to control them. If the biopesticides have some effect, then that would be a useful addition. But I'm not aware of a lot of research in that area. Have you looked at applying azoxystrobin midway through the crop to extend control? So no, we didn't, but I think there's previous work by Nitsan and Cummings and Johnson in the US, maybe mid to late 2000s, showing that a single foliar application about 35 to 60 days after planting reduced disease. So I think there is some information on that, and that it was effective in control, but that wasn't in combination with an in-furrow treatment. It was just a foliar treatment. Does black dot increase during the storage period? Yes, it can increase during the storage period. I think possibly less than silver scurf, but certainly it does increase, and by manipulating storage conditions, which I'm by no means an expert on appropriate storage conditions, you can reduce the levels of black dot development. How can you tell if you have clean seed if visually you cannot be sure of tuber surface infection? You can't. So some tubers are contaminated with inoculum, but you can't see anything visually. And that's the point about if you have a lot of disease on the tuber, very visually observable, it doesn't actually mean that you'll get more disease on the progeny tubers. So it is, without doing DNA diagnostics, you cannot be sure that you have clean seed without inoculum of the blemish pathogens. What is the easiest method of detection of these pathogens in Africa? Well, I think the methods of detection are the same visually, as we've just talked about the pros and cons of that, and using PCR methods. Quantitative PCR gives you a measure of risk. A conventional PCR would tell you whether the pathogen was present or not. What are the main resistant varieties in the UK against black dot and silver scurf? I'm not sure I can answer that question off the top of my head. And actually, there aren't always reliable disease resistance ratings for current varieties. So we would have some information on the varieties that we tested maybe 10 years ago. We've not tested them since. So I'm not sure I can answer that question easily. Have you done similar work on seed inoculum and soil inoculum on soft rot, pectobacterium, etc.? Not personally. I know there's work on seed contamination with pectobacterium. Not sure about soil because I'm not sure how detectable pectobacterium is in soil and how long it survives. That work ought to be possible, and certainly there's plenty of research on thresholds and levels on potato seeds. I'm working on a protocol to evaluate black dot on the stems of potatoes for my thesis. Do you have any ideas on how to do this? Well, I suggest that you send me an email and we'll discuss that in more detail. There are ways to do that visually and using PCR techniques. And there is some previous literature on development of black dot on stems. Which of the microorganisms are more efficient to control Colitotricum? I think I've covered that in terms of the question on biological control. Should cold stores and boxes be fumigated with a fungicide type disinfectant rather than clean to reduce silver scurf incidence? Oh, now you're testing me with that one. There is quite a lot of work from Stuart Whale, maybe 20 or even 30 years ago, about disinfectants and fumigation and store hygiene, comparing different disinfectants in store and different methods. So I can't tell you which disinfectant to use, but certainly store hygiene is very important in reducing silver scurf incidence, and that has been shown, and I'm sure there are references and grower leaflets that describe that. So I think the answer is yes. I'm not quite sure what the disinfectant is, particularly depends where you are. So comment, foliar azoxystrobin was tested in trials in Scotland and is effective, but I've no idea what the mechanism is. So yes. So as I mentioned, we did in furrow tests in the US, made foliar applications. Phil's suggesting that it was also tested in Scotland and was effective. So what is the threshold for black dot sampled in the soil that is considered a high level or a level that may cause economical damage? So I'm going to refer you to look back at the presentation and look at the graph of 125 soils showing the low, medium and high risk categories, and just look at the scale along the bottom of that graph and you'll be able to see what the low, medium and high categories are. Is bacterial wilt an economic disease in the UK? No, it isn't. I think I've got to the bottom of the questions there. So I think, unless there's any more coming, which I don't think, I think we'll draw to a close there. And thank everybody for attending and end the webinar there. Thank you.